Today we will talk about the coronavirus testing through real-time RT-PCR. The first step is actually the sample collection. How we get sample? We get sample through the nasopharyngeal swab or oropharyngeal swab. We put the swab inside the nasal tract or the oral tract, and we rub it with the walls of the nasal cavity, or in case of oral cavity, when we are talking about oropharyngeal. So after taking that swab out, we immediately transfer it into a sterile tube, which is already containing a viral transport material. Actually, this testing is based on the PCR procedure, and we know that the standard method PCR actually is a polymerase chain reaction. In case of PCR, we know that we get million copies of the specific fragment of the DNA rapidly. We know that the corona is a RNA virus, so it contains not DNA but RNA genome, and this RNA molecule is extraordinarily long fragment. So if we talk about detection, that how we detect it, for the first the detection is actually based on the actually first we have to extract it RNA, and then we go for PCR. The RNA is actually converted into the cDNA by reverse transcriptase. The RNA is actually that reverse transcriptase makes a cDNA, that is complementary DNA, and then that DNA is amplified by the PCR. So to detect corona, RNA must be converted into the cDNA sequences by the reverse transcriptase. So if we see, we can conclude it like this, that newly synthesized RNA is amplified by standard PCR procedures. This approach is universally known as RT-PCR. So if after collecting sample, if we come towards the protocol of the RNA extraction, we should know that the RNA extraction can be done by multiple type of commercially available RNA kits, using RNA kit. So to extract viral RNA by commercial kit, the sample is first added to a microcentrifuge tube. When we transfer our sample into a microcentrifuge tube, then the next step is addition of a buffer, which is known as lysis buffer. This lysis buffer actually, after addition of this lysis buffer, we vortex our microcentrifuge tubes and then incubate it at room temperature. After vorticing and incubation, we see that lysis buffer actually has caused our viral RNA to come out of the virus. So because it has a denaturing nature, this lysis buffer has denaturing nature. So under these denaturing conditions, the lysis of virus occurs. After this lysis, the next step is purification, which is actually carried out by using a purification spin column. Actually, we load it, then we go for centrifugation, and then we go for the solid phase extraction. Why we are calling this a solid phase extraction? Actually, the scene is, I should say, I think so. The case is stationary phase consists of actually in this solid phase extraction, the stationary phase, it consists of silica matrix. That's why we are calling it stationary phase extraction. The RNA molecule, it will actually bind to that silical gel membrane, while the protein and the other components, they aren't retained. So in this way, when we go through the solid phase extraction, our RNA, it will be retained, but the impurities, they will not be retained, the undesired components. After this purification or solid phase extraction, we go for the centrifugation again of that spin column. After that, after centrifugation, we place it into a clean collection tube and filtrate is removed. After that filtrate removal, we add washing buffer, and then we go for again centrifugation. As a result of that centrifugation, the impurities, they will get removed. When the impurities they are get removed, we again we actually go for, after washing, column is placed into a clean microcentrifuge tube. Then we add elution buffer. Now this elution buffer, after addition of this elution buffer, we go for centrifugation. Now this elution buffer actually will cause removal of viral RNA, and the RNA we will get up and that viral RNA is actually required. And in this way, in this last step, due to the addition of the solution buffer, and finally which we carried centrifugation, we will get at the end our pure RNA of virus. So in this way, RNA extraction is completed. After getting pure RNA, we move towards PCR, which is the next step. This is done. Actually, first we will have to do the preparation of PCR. Before we carry this process, the things we need, they are reverse transcriptase. I have written here RT, DNTPs, forward primer, reverse primer, Chapman probe. This probe will actually help in detection, and DNA polymers. The first thing we will have to do after getting all these components is we will add RNA template. Through these all components we will make master mix. When we will prepare a master mix of all these components, we will add template. After adding template, we will put that into the we will transfer master mix into the samples, and then we will go for the PCR. In PCR, the first step is reverse transcription. That RNA, we will get its cDNA, that is complementary DNA copies. The targeted sequences are RPD, RDP, RGRP gene, E gene, and N gene. And the second step after that reverse transcription is initial denaturation of the RNA DNA hybrid, which is actually formed as a result of reverse transcription. That that RNA DNA hybrid, in second step, it is initially denatured, which requires activation of the second step actually requires activation of RNA DNA polymers and inactivation of reverse transcriptase. Now the PCR, we know that it is actually the series of thermal cycles which have many denaturation, annealing, and the extension steps. We know that denaturation is actually carried out at 95 degree, while annealing is carried out at kind of low temperature, that is 58 as compared to the denaturation. And extension, its temperature depends upon which type of DNA polymerase you are using, DNA polymers they are being used. So in this way, we will finally after many cycles, we will get multiple copies. After denaturation and annealing and extension, many steps they will actually occur in case of PCR cycles. And at the end, we will get multiple copies of the DNA. And that PCR machine, it has actually camera. That camera will detect CCD camera. It is known as CCD camera. That camera will actually detect all that probe excitation light, and as a result of that light, it will give a graph, and that graph will show how the things are working inside, and we will come to know how many copies they are being produced. So in this way, we come to know all these things. Thank you for watching this video.